Okay, so we have seen how the female gametophyte was developing. Now we'll see how the archegonium developed. Okay, the archegonium, it will originate at the apex region of the female gametophyte. If let's say this is the female gametophyte, the archegonium will originate here at the apex. Okay. So each archegonium develops from a single superficial cell. As I said, the initial cells are always in the superficial part. And then this cell will divide periclinally to form a primary cover cell and a central cell. Okay. There it's written primary cover cell and a central cell. Then that primary cover cell will follow two vertical divisions at right angles to each other to form four neck initials. Let's see if it's there in the diagram. Okay, yeah. See, the primary initial, the archegonial initial cell at the tip of the mega gametophyte. Then it divides this way periclinally to form primary cover cell and central cell. Then that primary cover cell will divide to get neck initials and the and then on further division we will get more neck cells, and then the central cell will divide into two cells. This one divides into two cells, that is the primary neck canal cell and the primary venter cell. Then from that primary neck canal cell we will get the neck, and from the primary venter cell we will get the egg. Okay. Okay, so we saw in the diagram the primary cover cell divides to form four neck initials, and then again they will divide to get 8 neck initials. Then the central cell will divide to form the outer primary neck canal cell and the primary venter cell. Then from the neck canal cell we will get the neck, and the primary venter cell we will get the egg. Okay, sorry. The neck canal cell, okay, the primary neck canal cells does not divide further so that a neck canal cell is formed, but the venter cell will divide to form the ventral canal cell and the egg cell. Okay. So that is enough just from the diagram. Now let's see on the fertilization. Fertilization takes place after the megasporangium gets settled on the substratum, but in some cases it occurs while the female gametophyte is still within the sporangium. The biflagellate sperms are liberated, they swim to the archegonia through the water and fertilize the egg to form the zygote. Now the zygote is the new sporophyte. Okay, after fertilization the diploid sporophytic generation, that is the zygote, is established. The first division of zygote is transverse, we get an upper cell, that is epibasal, which develops into a suspensor, and the lower cell which will develop into the embryonic cell. Okay, the embryo is endoscopic, that is, it will direct it towards the base of the archegonium. Embryonic cell will divide into two vertical walls at right angles to give us a four-celled embryo. Then a short apical cell with three faces is formed because of oblique divisions in one of the embryonic cells. And then the embryo proper will undergo a 90-degree turn to its left. Then we will get the first pair of leaves on the shoot apex. So eventually, what is this? A foot is formed on the lower side of the embryonic tissue, and the primary root is developed between the suspensor and the foot. Then the young sporophyte will come out from the gametophyte tissue, that is from the megaspore, and it will continue to grow. Its shoot will continue to grow and the root will also grow. Okay, here, so you can see the embryo divided, no, into an embryo initial and a suspensor initial. The suspensor initial will give rise to this suspensor. Then again the embryo will divide this way to get 2-celled, and then it will divide again to get 4-celled. Then it will keep on dividing until it produces a foot, and in between the foot and the suspensor is the rhizophore. Okay, rhizophore is the root-like structure. I think, yeah, we have done about the rhizophore. Now let's look how it approaches seed habit. This sentence means, how is the Laginella similar to the upper plants which produce seeds? Okay, because number one, it is heterosporous. It produces 2 types of spores, okay, a male and a female. So even in higher plants, you have your androecium and your gynoecium. And then remember, the megaspore is reduced to 1 out of the 4. These 3 will degenerate. So even in higher plants, the 3 degenerate, only 1 is left. Then the megaspore is retained within the megasporangium. So just like the fruit in the upper plants, they are developed directly in the flower. No, so here also the megaspore is retained in the megasporangium, and fertilization will take place in the same place where the male and female plants are there. Okay, but also some scientists are saying it is not a true seed because first they say it is a true seed because number one, heterosporous, then only 1 megaspore is remaining, then the fertilization and development of the gametophyte takes place in situ. Okay, but they say it is not a true seed because the wall of the megaspore is dehiscent, okay, and it doesn't have the protective integuments like in higher flowers, higher plants. Remember, you have these integuments, okay, these side integuments, but here the spores are exposed. Then the retention of the megaspore permanently within the megasporangium has not been established, means we are not sure, okay, whether it's really retained or not. Then there is no complete fusion between the megasporangium wall and the megaspore. Okay, you have seen, in your practical, there is no complete fusion. Okay, so usually in the higher plants, these are the integuments, and if this is the egg, it is completely fused inside. Then the direct access of the sperm to the egg. Okay, this is not what a true seed is. A true seed is, remember, the pollen grain will germinate into a pollen tube and then it will travel along the style to finally reach the egg. But here directly, those biflagellate sperms will fuse with the egg. Okay, that is inside the neck cells. These are the neck cells. Okay, so we are done with this chapter.